Hello and welcome to this video on the complement system. This is not a new age self-help cult, but a part of the immune system. Its role extends beyond immunity, but hemostasis and organ development are only some of the features and functions it has. Complement has a big role in immunity. Unlike most parts of the immune system, complement is not cellular. It is instead a series of protein structures that synchronize through a cascade. This event is unsurprisingly called the complement cascade. The complement cascade is part of the innate immune system. That is the part of the immune system that is always active and able to handle many different pathogens. The downside to this is that while it is very versatile, it's not as efficient at handling threats when you compare it to the adaptive or acquired immune system. As this is part of the innate immune system, complement exploits conserved antigens on pathogens. These are sites that act as an indicator of what the thing is attached to. Is it a pathogen, your cell misbehaving, and so on? These all have antigens that can be used by the immune system. Your own cells indicate normal function, therefore the immune system need to look into them. Viral infections often present indicators in the form of an antigen on the surface of the cell. This allows the immune system to know what's happening. Bacteria and other pathogens of that type also have antigens. All of these things are indicated on the surface. As these are easily identifiable and relatively consistent across evolutionary time, the utilization has been conserved. If you look at complement from a physiological and anatomical point of view, it's made up of about 30 different proteins that you can find in blood plasma. They make up a tiny fraction of what is in your plasma. Interestingly though, when you take plasma and break it down into what it makes up, as far as the globular content goes, it makes up 15%. That's a surprisingly large amount. Given that it makes up a very large portion of what's in plasma, the way complement acts shouldn't be surprising. Fundamentally, complement recognizes anything that isn't protected from complement, and then attacks it. Your own cells will carry indicators in the form of an antigen that expressly tell the complement not to have anything to do with it. Anything that doesn't carry these indicators will become a free-moving target for complement to start attacking. This can include anything from foreign materials, damaged cells, and pathogens. All these begin the complement cascade. If we're talking about the early stages of an infection, this is where the complement cascade is most pronounced and in many ways most useful. Before your acquired or adaptive immune system can be rallied and have the full effect that it's going to have, your innate immune system is there. It slows down the pathogen and begins dealing with it immediately. Complement is part of this system. As the pathogen grows and becomes present, your body takes damage, and these activate up to 3 different pathways for complement. The first is considered the classical pathway, and this involves the binding of a particular structure called C1q, the first protein in the cascade. This attaches itself to the pathogen surface, often to an antigen site. This same system can also be turned on by the adaptive immune system. The adaptive immune system can produce antibodies, and the antibodies can get C1q to bind to them. When you look at this, you can see how complement acts as not only part of the innate immune system, but also as a bridge between innate and acquired or adaptive immune systems. While the classical pathway is the most commonly seen way for this to happen, and that is an antigen-antibody complex, you can get the alternative pathway, which is one of the others. These involve a protein called C3, or spontaneous complement component. These bind to foreign materials, pathogens, or damaged cells. The third and final pathway is called the mannose binding lectin pathway, and these also use the same C3 protein. This mannose binding lectin pathway is a good example of a conserved way that the immune system can recognize a foreign pathogen. No matter which of these three you're looking at, the same event occurs initially. That is, you get the C3 convertase cleaving the C3 to create the active version. This is C3a and C3b. These then further cleave other molecules, which create activation events for those. C3b also binds to the pathogen. This means that the pathogen now not only has a protein hanging onto it, but it's a much easier thing for the immune system to find, as you've got this indicator saying this is a foreign pathogen. That is just the first step in the complement cascade. C3b then binds to factor B. Factor B is then broken down to form factor BA. This then becomes C3b2BB, which is a protease, and this cuts C5 into C5b and C5a. These then also bind to C3b. C5a is important in that it is a chemotactic protein. That is, it draws in the immune cells needed to deal with whatever it is attached to. This is particularly useful since your immune system is spread throughout the body, and it may take some time for the immune system to find the pathogen. These signals help to draw them in and tell them exactly where they need to go. C3a and C5a also have another phenomena. This is they can cause degranulation, specifically of mast cells. These are the same cells involved in asthma. And much like with asthma, where you have constriction of vessels, you see constriction of blood vessels. This is useful if you want to prevent the pathogen from escaping. Once you have C5b active, you get something called the membrane attack pathway initiating as well. This is a raft of proteins, specifically C5b, C6, C7, C8, and polymeric C9. These together create the membrane attack complex, and it is the final form of the complement cascade. What happens here is it all forms together and turns into something very much like a tunnel. The tunnel punches through the membrane of whatever it's been attached to and allows for the movement of water into the pathogen. The pathogen then swells and lyses. As you can imagine, this kind of damage, if left unchecked, would very quickly and very unfortunately destroy your body. This is why the complement system is regulated fairly tightly. You don't want the feedback from this, which is a positive feedback loop, continuing to promote complement activity and the rest of your immune system. Imagine if you became sick from influenza, and all the symptoms you experienced would never go away despite the fact that you no longer were infected with influenza. That is very much what would happen if complement was not regulated. The way it's regulated is by a number of inhibitors. Most of these are there to try and avoid the issue of the early stages of the complement cascade being kick-started. Some of these are there primarily to stop later stages, such as C4b and C3b. By blocking those two steps in particular, you can more or less stop the complete assembly of the membrane attack complex. This helps then in reducing the inflammation and other immunological responses that would further amplify the effect of the complement cascade. Both C4b and C3b are convertases, and they're there to help other stages get started by breaking them down into the necessary components that would then be functional. We've spoken in other videos about the role of things like the B plasma cells. These produce antibodies that then bind to antigens. Research indicates that the complement system has a role to play in the functionality of these B cells, particularly that when you have the adaptive response, that is, the body's ability to find the right immune cells with the right antibodies and other cells, you have a better response. When you remove C3 in particular, which is an important part of the complement cascade, you don't get that response anywhere near as well. It greatly diminishes the efficiency of the adaptive immune response. Complement doesn't come from nowhere. It's produced by host cells. There are a variety of cells in the body that can produce the various components. C3b being one of the more tightly controlled ones that can be able to bind with your immune cells. There are a variety of receptors on the surface of the lymphoid cells, these being your immune cells. When you have both a combination of C3b and another cofactor, you find that these cells get activated. This can then change the structure and shape of these cells or receptors. This then allows them to change format or function. We've mentioned about the immune system and B cells in particular. More novel research indicates that C3 is an important actor in the antibody generation, that is, those B plasma cells that make antibodies. In short, antibodies are made by cells, and you get a random collection of cells that make a random collection of antibodies. The body has to find the right B cell with the right antibody in order to produce antibodies that will bind to a given antigen. This is why it can sometimes take a little while for the immune system to begin producing the right antibodies to a disease that you've not encountered before. In order for this to be done more efficiently, complement, and in particular the C3d, helps to attach itself to antigens, and this localizes the pathogen in an area where lymphoid cells can begin breaking it down and taking away the various parts of it that then allow the body to find not only the antigen, but then the right B cell which will make the right antibody to this. Later on, C3d, which is attached to these, plays yet another role in ensuring that your immune system retains a memory of what your body has been exposed to. This is through the B memory cells. Binding of the C3 to the right receptor on the B memory cell is essential. When the B cell encounters the antigen, antibody, and C3 complex together in the dendritic cells, this is where you get the necessary processes, not only for the formation of the B memory cell, but also the B plasma cells. Evidence indicates that this drastically lowers the requirement of adjuvants for the activation of B cells. Some evidence says it's 1,000-fold, and others up to 10,000-fold. We spoke very early on this video about how complement and other parts of the immune system rely on highly conserved indicators, and particularly antigens. Well, not every pathogen is going to play nice, and while the arms race on both sides has been mostly to the advantage of the human immune system, some pathogens have found ways to deal with complement one way or another. Some of these use the same regulators on the pathogen membrane that the human cells use. This allows the inactivation or retardation of complement. Bacteria, viruses, fungi, and parasites all seem to have some semblance of this, and different species more than others. The way these work is to prevent C3 convertase from functioning. This means you neither get C4 nor C3b. This means you don't get the complement attack complex being generated. Of course, not every pathogen has this, and as a consequence, it is still an effective part of the immune system. Another way bacteria and other pathogens have been able to thwart the complement system is that they create molecular platforms with high affinity. This means complement will attack that, but leave the microbe itself relatively unaffected, other than having to eventually replace that part of the cell. Parasites, fungi, bacteria, and viruses still have a functional body and therefore they can reproduce. It's much easier to fix a small part of the cell that doesn't have a functional purpose other than protection than it is to have the entire cell be destroyed. Complement is not necessarily essential, but it does play a not insignificant, if not central, role in many of the functions of the body. Particularly, it is the bridge between the innate and adaptive immune system in some ways. It also plays a big role in that innate immune system. It can, in its own way, help to support all the other cells in what they do without having to be a cell. It ensures support for B cells being almost the backbone to the adaptive immune system. Of course, complement, if there's not enough of it, will cause you harm because of this. But having too much complement or overactive complement can also cause harm, as can any overactivation of part of the immune system. Complement also has roles to play in ensuring hemostasis, blood clotting, side effects to certain medications, and there's a raft of other things that happen. But overall, this is a reasonable overview to what is happening with complement, what it does, and to a degree, why it does it. In the future, we'll provide a video explaining some of the interactions that are happening at the moment between the complement system and different drugs. We've already explained some of what you might see here in our video on blood clotting, or specifically the drugs used to treat blood clotting, these being warfarin and heparin. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.